Every pharmaceutical tablet or injection we produce carries a silent promise that it is safe for the patient who relies on it. But during drug manufacturing, tiny amounts of unwanted chemicals can sometimes form. These are called genotoxic impurities, and even extremely small amounts can potentially damage DNA and increase long-term cancer risk. Because of this, pharmaceutical companies must carefully identify, monitor, and control these impurities throughout the drug development process. In this video, we'll explore what genotoxic impurities are, where they come from, how regulators control them, and the advanced analytical techniques manufacturers use to detect them. Let's begin with the basics. Genotoxic impurities are chemical substances that can interact with DNA and cause mutations or carcinogenic effects. These compounds are particularly concerning because they can cause biological damage even at extremely low concentrations. In pharmaceutical products, the acceptable levels are often measured in parts per million or even parts per billion. Some common examples include nitrosamines such as NDMA, epoxides, and certain azo compounds. Because these molecules can potentially alter genetic material, regulatory authorities require strict monitoring and risk assessment to ensure patient safety. Genotoxic impurities can appear at several stages during pharmaceutical manufacturing. One common source is chemical synthesis. During drug production, reagents like alkylating agents or certain reaction intermediates can unintentionally generate mutagenic compounds. Another source is degradation. Exposure to heat, light, or humidity during storage can sometimes break down drug molecules and produce reactive byproducts. Finally, impurities may also originate from process residues, such as leftover materials from earlier manufacturing steps, or even trace contaminants from equipment surfaces. Because of these possibilities, pharmaceutical scientists must carefully examine the entire manufacturing process, not just the final product. To manage these risks, global regulatory agencies have established strict guidelines. The most important framework is ICH M7, which provides a scientific approach for assessing and controlling mutagenic impurities in pharmaceuticals. A key concept introduced in this guideline is the threshold of toxicological concern, or TTC. This sets a general exposure limit of 1.5 micrograms per day for lifetime exposure. To support risk assessment, pharmaceutical scientists also use structure-activity relationship tools, often called SAR tools. These computer-based models predict whether a chemical structure may have mutagenic potential. Together, these regulatory frameworks ensure that potential genotoxic risks are identified and controlled early in drug development. Once potential genotoxic impurities are identified, companies follow a structured risk assessment process. The first step is identification, reviewing the entire synthetic pathway to determine where mutagenic compounds might form. The second step is evaluation, where scientists assess the potential toxicity using computational models, experimental data, and regulatory guidelines. Finally, the third step is control, which involves designing strategies to either eliminate the impurity, reduce its formation, or monitor it at safe levels. This step-by-step framework helps manufacturers ensure that any potential risk is carefully managed before the drug reaches patients. Detecting genotoxic impurities is extremely challenging because they exist at trace levels. To achieve the necessary sensitivity, pharmaceutical laboratories rely on advanced analytical technologies. One of the most widely used techniques is LC-MS, which combines liquid chromatography with tandem mass spectrometry to detect non-volatile impurities with extremely high sensitivity. For volatile compounds, scientists often use GC-MS, which is particularly effective for detecting impurities like nitrosamines or formaldehyde. Another powerful tool is high-resolution mass spectrometry, or HRMS, which allows researchers to identify unknown impurities by measuring molecular masses with exceptional precision. Together, these techniques allow detection at sub-ppb levels, ensuring even the smallest risks can be identified. Developing analytical methods for genotoxic impurities comes with several technical challenges. First, the detection limits are extremely low, sometimes between 0.1 and 5 parts per billion, which requires very high analytical sensitivity. Second, there is the issue of matrix effects. The complex chemical composition of the drug substance can interfere with analytical signals, making accurate detection difficult. Another challenge is stability. Some impurities degrade during sample preparation, which means laboratories must handle samples carefully using inert conditions. Because of these issues, analytical methods must undergo strict validation to confirm their accuracy, precision, specificity, and robustness. In the pharmaceutical industry, the most effective strategy is not just detecting impurities, but preventing them. One approach is designing safer chemical processes, where chemists choose reagents that are less likely to produce mutagenic byproducts. Another common practice is purification, using techniques like crystallization or solvent extraction to remove impurities during manufacturing. Companies are also increasingly adopting process analytical technology, or PAT, which enables real-time monitoring of impurities during production. These strategies help manufacturers detect deviations early and maintain consistent product quality. The importance of genotoxic impurity control became very clear during the global valsartan recall. Trace levels of nitrosamine impurities were detected in certain batches of the drug, leading to a worldwide recall that cost the pharmaceutical industry over $1 billion. This event demonstrated how small analytical issues can have massive consequences, not only financially, but also in terms of patient trust. On the other hand, companies that proactively manage genotoxic risks often experience faster regulatory approvals and fewer delays in new drug applications. In today's regulatory environment, proactive impurity control is both a scientific necessity and a business advantage. Looking ahead, the future of genotoxic impurity control is becoming increasingly technology-driven. Artificial intelligence and machine learning are now being used to predict mutagenic risks even before synthesis begins. At the same time, advances in analytical instrumentation and real-time monitoring systems are enabling continuous detection of impurities during manufacturing. These innovations are transforming genotoxic impurity control from a reactive process into a predictive and preventive strategy. Ultimately, these advancements will help pharmaceutical manufacturers deliver safer medicines while meeting the highest global standards of quality and compliance.